Hi all, in this video we're going to look at importance and exportins. Here are some keywords we're going to know or learn through this video. Nuclear, nuclear localization signal, RAN, and export signals. Okay, so let's dive in. So the nuclear pores, as you know, are channels that allow molecules like mRNA, proteins, etc., that need to enter the nucleus or exit the nucleus actually. Okay, so it's like a channel that allows things, let's put it that way, to go through in and out of the nucleus. So if we look at the top view of this nuclear pore, we'll observe an octonomer signal, symmetry, right? And yeah, and then here over here are the cytoplasmic filaments. So that's this, yeah. The octonormal symmetry is what I've tried to show in this with this purple, sorry, no, yellow cylindrical, not really, but things. Okay, yeah. Yes, and then here are the cytoplasmic filaments. Okay, and these cytoplasmic filaments help with docking. Okay, so let's look at more into the nucleus. So if we look at this picture right here, what we notice that if we take a zoom in like of the nuclear pore, we'll see this mesh work right here that kind of looks like spaghetti, but not really. This is called the FG domain. Okay, it's made of phenylene, yeah, and glycine. So those amino acids, and they're hydrophobic too. Okay, so um if a protein or if whatever needs to enter the nucleus has this address, has the nuclear localizational signal, and LS, that's what it's called. Okay, so if it has a nuclear localization signal attached to it, then it'll go through, interact with this FG domain. Okay, I'll show this in the next slide, and it will be able to pass through into the nucleus. If it doesn't have it, then obviously it can't pass through the nucleus. Okay, because the nucleus needs to regulate what goes in and out of the cell. However, really small particles that don't really need that can just diffuse through the nuclear membrane, or yeah, basically. Okay. Okay, so in this next slide, we're going to look at step-by-step process of how larger molecules such as proteins, mRNAs pass through the nucleus. Okay, but in this case, we're going to look at a protein. So we have our protein right here. No, right here. Okay, so this is our protein. These things, this right here, this and this, are called our importance. More specifically, this would be our alpha subunits, and this is our beta subunits. Okay. Now, these two importance are going to bind to this protein, right? As you can see in this picture, they bind to attach to it, and together that's going to move through the nuclear pore complex. Okay. So in this slide, as you can see, they have kind of passed through the nuclear pore, and they were able to do it successfully because the protein has this nuclear localization signal. Okay, so when they enter the nucleus right here, they're going to meet a friend called RAN GTP. RAN GTP. This is important because it's going to break down, dissociate it. So kind of like break it down like this. Okay, so this protein, our protein, is going to be free to go to do whatever its job is. Maybe it's to activate transcription, whatever its job is, it's going to go ahead and do it. And why the beta subunits, let's put it up here, and the alpha subunits are going to be busy with some other friends. So RAN GTP is going to bind to the beta subunit, while a protein called exportin is going to bind to the alpha subunit, as you can see here. Okay, I'll just use this dot to put the alpha. Okay, so the exportin is going to bind to the alpha subunit, while GTP is going to bind to the beta subunit. And together they're going to move again through the nuclear pore outside to the cytoplasm. So this is our cytoplasm right here. Cytoplasm. Okay, perfect. One thing you should know that these exportins also contain a nuclear export signal. Okay, so that means that okay, we're going out because you also need those two. Okay, so when we get to the cytoplasm, here's what's going to happen. RAN GTP is going to be hydrolyzed right into RAN GDP. So this is GDP. GDP. Okay, so it's inactive. It can't perform any function basically in this state. Okay, and because a consequence of that is that our beta subunit is going to dissociate from it. Okay, and our exportins, when they get to the cytoplasm, they're going to obviously let go of the alpha subunit. Okay, and together this is going to go back into the nucleus, and then so is this. Okay, before we get into that, you should know that what drives this hydrolyzation, what allows this process to go, is basically done by um a done by a protein called RAN GAP1. Sorry, that's N. RAN GAP1. Okay, and I'll just, for the sake of clarity, move this away. Yeah, just move this away so you don't confuse this with something else. Okay. Yeah, so RAN GAD1 kind of like drives this hydrolysis of from GTP bound state to the GDP bound state. Okay, good. And the purpose of doing that is to create a high concentration of RAN TP in the nucleus and a lower concentration outside. Okay, but I will explain that further in the next slide. But this picture just shows again the hydrolysis reaction, okay, from an active state right here to an inactive state right here. Okay, just to kind of like make that a bit more clear, this is the active state of RAN GTP right here, and this is the inactive state for RAN GTP. So active, inactive, basically. Okay, so in the next slide, so as you can see, it's in its inactive state right now. We're going to make this active by adding GTP to it, okay? And what performs this reaction is a protein called RIG. So R C I. Okay, so it's going to drive this reaction so it becomes active. And in its active form, it can go ahead and interact with, whatever the protein, it's going to go ahead and interact right here with our complex once it enters the nucleus, of course. Okay. Okay, and that is it for this video. Thank you, and please subscribe.